Thank you for the opportunity to present here today. I appreciate all of those conversations that I've had, and I've tried to rearrange my talk a little bit during the day to meet those, so I may skip over some slides, but all of those will be available to you to look at later if I go by something too quickly. Okay, so I'll talk a little bit briefly about the NIH Common Fund Metabolomics Program, sort of what the resources are and how you can get to those, and then several applications in metabolomics that I thought may be interested to people who are doing nutrition research. Okay, so there are 6 NIH Common Fund Metabolomics cores in the US, and they're at UNC Chapel Hill, Kentucky, Michigan, Mayo Clinic, Florida, and UC Davis. And all of these cores were created by the NIH Common Fund program to promote clinical, translational, basic science research doing metabolomics. And they're very collaborative, and if you want to access any resources, contact anybody who's at those, and you can have these slides later to see. We also have a resource that's called Metabolomics Workbench. There's a website for that. You can just Google Metabolomics Workbench and you'll find a link to it. And it tells you all about many of the resources that NIH has put available in the US and types of studies that we're doing, et cetera. Okay, and there's many hands-on training materials, and you can find those described. There's web-based learning materials, and you can find all that at the Workbench. Okay, so I was supposed to focus on applications, and so I just wanted to start off by saying there's many studies that have shown metabolomic signatures that correlate with gender, race, age, ethnicity, drug use, chemical exposure, stress, weight status, mental health status, blood pressure, many disease states, behaviors, nutrition, gut microbiome, and I'll show you some work that I've recently done on the correlation with blood types. So we know that it's successful in being able to reveal biomarkers that are associated with the types of things that we're looking at in health, in studies of health and wellness. And I won't say much about technology because David did such a nice job describing it, but there are many types of technologies that are needed to really get at what the metabolome is. And the nice thing is that those many technologies can be used on all types of biospecimens. They could be used on food specimens, but also on saliva, sweat, urine, serum, plasma, fecal matter, breath. And for a large part, our metabolites are very conserved across species and very conserved across the type of biospecimen. So we can use similar methodologies and then be able to make comparisons between studies. Okay. So one of the things that David was talking about is how you need many, many technologies to detect all these metabolites, and that's true. But no matter what technologies that we're using, we typically have a lot of metabolites or a lot of signals that we don't know what they are. And so one of the big challenges in metabolomics is synthesizing standards of the real unknowns or the unknown unknowns to try to understand more about what the metabotype is. So we know what we know, but we don't know what we don't know yet, right? So there's a lot of effort in doing that in the metabolomics community. So my course done a lot of applications. You'll see the research areas listed on the left, and the sample types are pretty extensive. And then the origin of those samples come from cells, from model systems, from human subjects, and at all ages, and you know, it's very applicable. So probably for about any kind of biospecimen you can get in any disease or state of health can be applied. So metabolomics largely involves in mammalian metabolomics or clinical studies largely involves this comparison of states of wellness and states of sickness to reveal biomarkers that can be more sensitive for the early detection of disease. So that's what a lot of the effort has been in, but also to understand mechanism associated with those disease state. And then in the area of nutrition, of course, we'd want to be able to identify what are the nutritional targets, like what could we do to supplement or to change diets. So I'll show a study that I conducted with Michelle Williams and Bazu Galay of Harvard University, where we looked for early serum markers of third trimester placental abruption. Placental abruption is a disorder during pregnancy. It occurs in 1% of all pregnancy and has a premature separation of the placenta before delivery. And it can be fatal to the mother and child. It's very difficult to diagnose. And so our goal for this study was to determine in serum in the second trimester before any pregnancy complications showed up, if we could predict who would have a placental abruption or not have a placental abruption. And so these studies were pulled from a biorepository, and we had all second trimester samples before any clinical complications, and the phenotype was the end of study, whether half the women had the placental abruption, half of them didn't in the third trimester. And I show this as an example because it worked. There were 9 metabolites that were significantly associated with the presence of placental abruption, and we did a targeted method for 188 compounds on this study that ranged carnitines, amino acids, amines, glycerophospholipids, phosphatidylcholines, and sphingolipids. And here you'll see the ones that had a p-value that was significant in significantly different between the cases and the controls of placental abruption. And then that data was modeled by logistic regression modeling to determine the probability of a placental abruption in the third trimester based on the serum biomarkers in the second trimester. And the gold standard right now for placental abruption is vaginal bleeding, but that doesn't occur until you already have that pregnancy complication and it's a little late, right? And it's not really telling us about mechanism, it's telling us that there's a problem. But we looked and we could see that our metabolites could predict more or about an equal number of cases before any complications in the second trimester serum than vaginal bleeding could do that happened much later. So it's beating that gold standard, which is really good. And if we use them in combination, we have a significant increase in that prediction. So, but mechanistically is what's important in this study. And what we were able to see is that the probability of the placental abruption was increased with an increase in acylcarnitines and a decrease in phosphatidylcholine. And these metabolites are closely related at diacylglycerol, which is transformed to an endocannabinoid called 2-AG, and 2-AG is converted to prostaglandin glycerol esters. So we know that PTGS2, which converts 2-AG to these prostaglandin glycerol esters, is downregulated in the aging placenta. And so our observations suggest that this PTGS2 may play a role in the pathogenesis of abruption. We also know that low-dose aspirin is given to some women who are at high risk for adverse pregnancy complications. And PTGS2 is a major target of non-steroidal anti-inflammatory drugs, including aspirin. And we also know that choline plays a critical role in pregnancy and women's health, and there's lots of information that's been shown on that. And we have other evidence of the importance of choline in lowering concentrations of an FMS like tyrosine kinase 1, which is known to disable proteins that cause blood vessel growth. So together, our studies are really showing that there's this importance of choline early during pregnancy. And we've known that it's been very important for the neonatal, the brain health, et cetera, but we believe it's really important in pregnancy complications. And so we're now working on a study. We're running a validation study right now with a larger number of samples derived from from a biorepository, second trimester serums, to see if we can reproduce this result that phosphatidylcholine is decreased in women who go on to have a placental abruption. And we're also incorporating broader spectrum metabolomics methods to see what else that we can find. So at this point, we have identified choline as being very important in the case of placental abruption, and that's what we're working on. We've done some work with metabolomics and autism, and this was also published recently. It was in the Chinese Han population, where we did a training set and a test set of autistic subjects versus neurotypical subjects. And to make a long story short, because I know I could take the whole time just talking about this study, I want to say that we had 17 metabolites that were identified in our training set and 11 metabolites that were validated in the test set of about 100 of each subject. And sphingosine 1-phosphate and docosahexaenoic acid were both very important metabolites in distinguishing autism from neurotypical. And those two made a lot of sense. DHA is a supplement that's used in autism, often in autism subjects within the US, but was not being used in the Chinese Han population, and it was shown to be decreased in the autistic versus the neurotypical subjects, sort of validating that use within cohorts that we know of. And then we were also able to identify 9 other metabolites that were either increased or decreased. And this is a case now where we're taking the results from the human subject study and into an animal model of autism to determine if any of those compounds can be supplemented to reduce some of the behavioral phenotypes that are observed in those animal models. So it's reverse translational, right? Okay. So I wanted to give a couple of examples on predicting response to treatment now. So those other two were on, identification of a clinically relevant biomarker and gaining some insights into mechanisms. And now predicting response to treatment is a fairly huge area in metabolomics. So the first study, that's it. One of the first studies that we had done in human subjects was in an adolescent obesity and response to intervention study in collaboration with David Collier at East Carolina University. And he had obtained urine samples from adolescents participating in a 3-week healthy camp for overweight children. And I say healthy camp because it wasn't a diet camp, it was a healthy lifestyles camp, and teaching children how to eat right and motivating them to get out and exercise, not run, but to get out and canoe and, kayak and swim and, have fun. And so they did a lot of clinical measurements during this healthy lifestyle camp, and they also collected baseline urine samples and end-of-study urine samples. And the one part I'll talk about here is that at baseline, the branched chain amino acids were at lower levels in children that were responders to the weight loss program compared with children that did not have a positive response to the weight loss program. And by positive response, I mean a clinically significant reduction in BMI. And so this was interesting because there's been some other studies in adult populations that have shown that these branched chain amino acids at high levels are predictive of the development of diabetes. So we're seeing something consistent, that the children that aren't going to lose weight, or these high levels of branched chain amino acids at baseline in children who don't lose weight, clearly could be predictive of development of diabetes later in life if they don't get into a program that does. And it really, if we can validate these markers, it would really help clinicians be able to understand who to have in these programs for weight loss and who not to, because they don't want to be dealing with psychosocial wellness as well as BMI for children who aren't losing weight. Yeah, so that's kind of where I'll take that one now. We've also done some work with David Lozier at UNC Chapel Hill and diet and exercise programs. This one happened to be a diet and exercise program from the IDEA study, and it was looking at osteoarthritis. And we were able in that case also to see baseline levels of glycolate, hippurate, and trigonelline that were different in subjects who progressed or did not progress in the score of osteoarthritis. Again, clinically relevant in helping guide intervention studies. And also just to mention that some of the metabolites that we've seen in this study and other studies are microbially related and could really show with some further research, the importance of this gut microbiome and how is that related to metabolites that we see being important in the progression of disease. So with that, we actually did a study with Subhra Sagi on alternative strategies for preventing or delaying the initiation of dialysis by targeting the gut-kidney axis. And in this case, participants were given a capsule of a supplement called Renadil, and those capsules contain the different kinds of bacteria that are listed on this slide. And you can look at the details on these slides later, but I'll skip over some of the details now and say that we did the metabolomics analysis on blood that was collected at enrollment and up to 4 months following treatment with probiotics, and that some of the subjects had a decrease in the BUN, which is a good thing, right, following the probiotic treatment, while other subjects did not have a BUN decrease. And you can see that we derive what are the metabolites that are associated with this decrease in BUN. So that's the goal. So their goal, to understand that at baseline, what metabolites are associated with having a BUN decrease later, because that could be clinically relevant if you can validate to be able to understand who should get some kind of treatment or not get that kind of treatment. But also this helped us understand more about the mechanism of it. And this again came back to the choline pathway where the metabolites in red, and many of the metabolites that we saw that were either increased or decreased in BUN, or some people say that changed in the low BUN versus the BUN that didn't change, are related to this choline metabolism. And this makes a lot of sense because the kidney synthesizes osmolytes, betaine and glycerylphosphocholene, from choline to retain water and respond to kidney tonicity. Choline deficiency is associated with damage to the kidney and changes in the gut microbial ecology. There were a lot of metabolites that we saw were related to gut metabolism in there. Due to its metabolism in proximal tubular cells, others have hypothesized that elevated plasma choline may signal tubular interstitial dysfunction, a pathology which is even more highly correlated with chronic kidney prognosis than glomerular injury. Trimethylamine and dimethylglycine are known urea solutes, and metabolism of choline by the gut microbiome is a major contributor to their synthesis. And then the presence of dimethylglycine in the lumen has also been positively correlated with the presence of bacteria. And so those are sort of evidence, again, in this study that we have this importance of choline metabolism in response to probiotic treatment. Okay, so I'm going to talk about one or two studies quickly on model systems and then tell you about some of our recent blood type work. So in our foods, you know, there are many things that are ubiquitous in the environment that can get into our foods that have been characterized as endocrine disruptors. And we've been studying a number of those kind of foods, a number of those kind of compounds in foods that get into foods. Sorry about that. how did I say that? One of those has been phthalates, where we did studies in pregnant rats exposed to butybenzyl phthalates during gestation and looked at what kind of biochemical perturbations were caused during pregnancy, but also in the offspring that were born after the in utero exposure. And to make a long story short, we did see perturbations in the biochemical pathways of these offspring that were born to dams who had exposure to the phthalate only during gestation, compared with pups who were born to dams who were exposed to the vehicle alone. And these were at effect levels of butybenzyl phthalate that resulted in reduced anogenital distance, but they were also at what had been described as no effect levels at butybenzyl phthalate. So we do see the perturbations at these no effect levels. We did this study first in urine and then went back and examined testes, brain, and uterus. And there were notable differences in the testes of the male pups, but not in the uterus of the female, in response of the biochemical profiles in response to the treatment with butybenzyl phthalate. And the male and female brain, both of the pups, but not the dam, both also showed perturbations, which makes sense because butybenzyl phthalate is a male androgen, I guess that's, yeah. And I had worried about the perturbations in the brain, thinking that butybenzyl phthalate didn't cross the blood-brain barrier, but it didn't perturb in the adult, it only perturbed in the pups exposed in utero, and that could make sense because in utero the blood-brain barrier is not fully developed. So, and that's published if you'd like to see it. And we've gone on to do this work now. We've done some work with nanoparticles, arsenic, PAHs, done some recent work with BPA, and flame retardants during early life exposures, looking at these perturbations once the exposure has been stopped. Okay, so how much time do I have left? 5 minutes? Okay. So we have done some work on diet and the ovarian and serum metabolome. These studies were conducted with Sue App at Wake Forest University, and she had serum and ovary samples from monkeys that had been either on a prudent diet or a Western diet for 2 years. And this Western diet was a high in cholesterol and saturated fats, and the prudent diet, of course, was low in those. So we did both targeted methods and broad spectrum metabolomics, and were able to find perturbations in amino acids, acylcarnitines, biogenic amines that were significantly different between these two diet groups for serum and for the ovary extracts. And one of the things I wanted to comment on this study that was recently published is that the perturbations that we saw in the ovary were very similar to those perturbations we saw in the serum, which could mean that you could develop a serum-based profile for effects, adverse effects on the ovary, or ovary quality, maybe I should say. So that's sort of a future research idea. We've done some work on looking at antibiotic-mediated gut microbiome because we know antibiotics are getting into the food. This was done in collaboration with Martin Blaser at New York University Medical Center. This was a study conducted in mice using a non-obese diabetic mouse exposed to the antibiotic or exposed to vehicle. So that's the comparison. And in his model, which you've probably seen some of his work published if you're following this antibiotic literature, he sees type 1 incidence of diabetes was increased in the antibiotic exposed animals compared with the control animals. So Martin Blaser's group has done all the microbiome work related to this study, and they were able to show in metagenome analysis the antibiotic-exposed mice were enriched in lipid, amino acid metabolism, and reduced for butyrate. Wamal Pathmasiri and MyCore conducted an analogous study, not knowing what their results were. They were doing these simultaneously, And he showed metabolomics profiles between the antibiotic and vehicle-exposed animals are distinguished by amino acids, lipids, and butyrate metabolism. So it's very nice to have these two complementary approaches that are really pointing to the same pathways for perturbations by antibiotics. And just as a last snapshot of some work that we've been doing recently, is looking at the differences in the metabotype of blood types. So we know that blood types, this is pretty basic about, we have different blood types and they're different across different ethnicities. And I had started thinking about doing this work because people have shown metabolic profiles can be different based on ethnicity. So I had wondered, is that diet or is that something fundamental to the blood, right? So, and that sort of was the reason. So one of the papers that we recently published was on blood type biochemistry and human disease, sort of showing, what do people know about our blood type and our risk for disease? So we published that in, I think it was just earlier this year. So if we go back, we know that ABO blood type is located on chromosome 9, and that there's 3 main allelic forms, A, B, and O, and each responsible for the production of its glycoprotein. So if we know that the glycoproteins are different, because that's how we characterize blood type, right, then we can assume that the biochemical profiles of blood type are different. So it seems so logical now, but it's just before, should we really do it and invest in it? So, and there is some evidence that there's a correlation of blood type, not only with many diseases that we've summarized in our review article, but also with this H. pylori infection. And in that people with H. pylori infection, their stomach produce less intrinsic factor, which is needed for B12 absorption. So we know that there is this blood type and absorption, disease risk, and all these things that are related. So with that in mind, we did our first study, which was looking at serum from 40 white healthy males, ABO, A, and B blood types. And in the first analysis, I used an NMR metabolomics analysis, and we could get very, very clear distinctions of those blood types. And you can see in this figure, it just depicts what we called pool samples. We ran all the samples individually, but also pooled for each blood type for the O and the AB, which should be further apart if you think about genetically in the inheritance as it's shown in that diagram, and then the A and the B are sort of in the middle of that multivariate space. So there was some concern about reproducibility because, well, nobody had done this before. So we did reproduce it, and I did want to say it was reproduced on a different instrument by a different operator at a different time. And in the models that we developed, these multivariate models, the Q-square is 0.9 on the multivariate models, which is very strong. So to be able to say there is this difference, it's a very strong Q-square. So what I started doing was looking at, well, if I take one blood type and I analyze it against all other blood types, what are the signatures for that one blood type alone? And then, so we could get a signature for O, A, B, AB. And of course, with different technologies, right, you're going to get a different signature because, you'd have to be using every single technology in the world to get the right answer. And you'd have to know what all the unknowns are too. But this is a start on how we can do this. But it's like the next question. And then we also, just by the way, reproduced it with UPLC TOF-MS and with 2D-GC TOF-MS, just to make sure that it's not just one analytical platform that we're seeing it on. And we also used a lipidomics method. So this is pretty thorough investigation of the blood type now. And so the list of these analytes that differentiate these groups are growing as we do more and more methods. Okay, so last slide. And this is just to give a little tidbit of example of why I think blood type could be important as we talk about nutrition and we try to think about how to personalize nutrition. And I do want to say that I'm a physical chemist, and I had never heard about eat right for your blood type. So that's not where I was going with this from the beginning. And I just want everybody to know that I'm a physical chemist trained in spectroscopy. It was just an idea that metabolomics could differentiate by blood type, just because I knew that in studies where you had different ethnicities, that was something that was important, you know, that the metabolomics profiles could distinguish that. But I've been looking at these now in more detail, and this is just a few examples based on the GC-TOFA-MS. I have about 20 such stories. So one thing I wanted to say is that the branched chain amino acids were higher in AB blood type, and branched chain amino acids have been associated with increased risk for diabetes. And diabetes in epi studies or clinical studies has been shown to be at higher risk in non-O blood groups. So that's like one piece of information. Tyrosine, which is involved in the production of stress neurotransmitters, epinephrine and norepinephrine, it is taken as a supplement to fight off depression and boost dopamine levels. And tyrosine was higher in the AB blood type. And there are epidemiology studies that have shown that type O blood type has more depression and intense anxiety than the AB blood type. So this is sort of another little story. And then we have some stories on saturated fatty acids and heart disease and other such things as we go through each individual blood type. So the goal of our studies now is to look at what the blood type is, what the risk of disease is, and then what compounds are either increased or decreased in that blood type that you can metabolically associate with that risk for disease. So it's not just the pattern of change, but also can you come up with a mechanistic story like the tyrosine and neurotransmitters and the link to depression and anxiety. So that's kind of where we're going to that. And with that, I'll stop and thank my group at UNC. Thank you.
All right, and with that, if there are any questions for Dr. Wishart or Dr. Sumner, please use the microphones.